Picture a clump of human brain tissue, like a tiny floating cluster of cells.
Right, a cortical organoid.
Exactly. And picture it living in a laboratory incubator, just sitting there, suspended in this fluid for 5 straight years.
5 years. It's really a profound image when you think about it.
It is,
Yeah.
because it has no body, no eyes to see the sun rise or set. There's literally no calendar on the wall of this incubator.
No sensory input at all.
None.
Hmm.
And yet, down to its very DNA, this little floating cluster of cells knows exactly how old it is.
Yeah, the internal clock is it's relentlessly ticking away, regardless of where it is.
Which is completely mind-bending. The idea that isolated cells can keep time without a metronome, it gets at something so fundamental about who we are. I mean, human brain development is famous in the animal kingdom for being incredibly, almost agonizingly slow. Right?
Oh, absolutely. We call that extended childhood neoteny. Building a human brain, it isn't just about having the right materials. It's a meticulously timed manufacturing process. I mean, getting the exact right cells to emerge at the exact right time, that is the fundamental requirement for making our neural architecture function.
So it's all about timing.
Entirely. If a specific type of neuron shows up to the construction site, say, a week early or a month late,
The whole thing falls apart.
exactly, the entire scaffolding can fail.
Wow. Okay, so the stakes for this biological timekeeping are massive. And that brings us to the mission of today's deep dive. We want to figure out how this works in a dish without a physical calendar. And to do that, we really have to zoom all the way down to the molecular level.
We do, yeah. We have 3 really incredible papers from Nature to guide us through this.
Right, so let's start with the first one, a study by Faravelli and colleagues. They're the ones who managed to keep these human organoids alive for 5 years. Now, my understanding is that keeping a mini brain alive for half a decade is practically unheard of.
It really is. Historically, keeping neurons alive in a dish for that long has been, well, a massive hurdle.
Why is it so hard? Do they just run out of nutrients?
Kind of. What usually happens is that as the organoid grows, the cells in the very center get cut off from the nutrient bath.
Oh, because it gets too thick.
Right. Without a blood supply to physically pump oxygen into the core, that center becomes necrotic. It literally starves and dies from the inside out.
Ugh, wow.
Yeah, it's not great. But the Faravelli team, they optimized their growth conditions using something called an APM medium, which stands for activity permissive medium.
Activity permissive. Okay, what kind of activity is it permitting exactly?
Electrical activity. Neurons need to fire to survive. It's very much a use it or lose it system.
Oh, interesting.
Right. Standard lab broths keep the cells fed, sure, but they often contain concentrations of certain ingredients that actually suppress complex coordinated electrical firing.
So they're fed, but they're basically chemically sedated.
That's a good way to put it, yeah. The APM medium balanced the chemical environment, specifically things like calcium and potassium, so that the neurons could form active networks.
And fire on their own.
Exactly. They could fire spontaneously without wearing themselves out or creating toxic byproducts. And by keeping that network healthy, they extended the viability of these cells far beyond previous limits.
So they essentially built a better artificial life support system.
Pretty much.
And that let them observe something unprecedented, right? Like cells actually growing up over 5 years. The paper notes they saw years of ongoing transcriptional and epigenetic maturation.
They did, yeah.
Now, transcriptional makes sense to me.
Yeah.
You know, genes turning on and off as the cell gets older.
Yeah.
But how does epigenetic maturation function as a clock? Because when I hear that, I picture like counting the rings on a tree trunk.
Right. Tree rings are a really helpful visual for the passage of time, but mechanically, epigenetics operates more like microscopic padlocks on your genome.
Padlocks.
Yeah, so think of the DNA itself as the hardware of a computer. The actual sequence of letters doesn't change as you age.
Right.
But epigenetics is the software layer that dictates which programs run. The Faravelli team specifically looked at DNA methylation.
Which is what exactly?
These are tiny chemical tags, methyl groups that physically attach to the DNA strand, usually right on top of a gene.
And when a methyl group attaches, what does it actually do to that gene?
It basically creates a physical roadblock. When the cellular machinery comes along trying to read that gene, to build a protein from it.
It hits the padlock.
Exactly. It hits the methyl group and stops. The gene is effectively locked down or silenced. And as an organoid ages, these methyl padlocks accumulate across the genome in very predictable, distinct patterns. So by looking at exactly which genes are locked and which are open, researchers can calculate a highly accurate biological age.
Wow.
Yeah, and in this case, the DNA methylation age of those organoids tracked perfectly with the actual number of days they had spent in the incubator.
Okay, let's unpack this because that answers how they track time. But there's this experiment in the paper with a wild name, heterochronic chimeroids.
Oh, yeah. This is a fascinating one.
Right. And it tests if the cells actually remember that time. I know hetero means different and chronic relates to time. So what exactly were they mixing together here?
Well, they wanted to test the resilience of the cellular memory. Like, if you take an old cell and transplant it into a completely youthful environment, does it reset?
Oh, wow. Okay.
To find out, they took progenitor cells, those are the stem cells that give rise to the brain from older organoids, and they physically mixed them into a culture with brand new, young progenitor cells.
Just mash them together.
Yeah. They were looking to see if the chemical signals from all those young cells would convince the old cells to forget their age and just start over from scratch.
So if I can try an analogy here.
Uh-huh.
Earlier I was thinking about this like a factory worker getting transferred to a new assembly line, right? But based on the padlock idea you just mentioned, it sounds more like a computer installing a massive software update.
Okay, I like where this is going.
Like, if you take a hard drive that's already 80% through an installation, and you unexpectedly unplug it and plug it into a brand new motherboard, it doesn't start back at 0%.
Right, it does.
It checks its internal log, sees the padlocks are already in place, and skips straight to the final 20% of the download.
That captures the mechanics beautifully. The older progenitors, they did not reset.
Wait, really? Not at all.
Not at all. Even when bathed in chemical signals instructing them to take on an early developmental fate, they just ignored those external cues.
Because their internal software was already updated.
Exactly. Their epigenetic software was locked in. They entirely skipped the production of early-stage cells and immediately started generating the late-stage excitatory neurons they were scheduled to produce.
That is incredible.
They recorded the passage of time, and they fiercely protected that memory.
So the internal clock is perfect.
It is perfect.
But, a clock is ultimately useless if the alarm goes off and the person just stays in bed.
Right.
Are these cells actually getting out of bed and doing their jobs on time?
Oh.
Because when we look at our second source, this is a Nature study by Stauffer and colleagues. The daily routine of these cells seems to be a complete mess.
Yes, and this is where we have to separate the concept of knowing the time from the actual execution of the daily schedule.
Okay, explain that.
Well, Stauffer's team looked at mouse cortical organoids, and they asked a critical question. Is the cellular orchestra actually following the molecular metronome?
Oh.
And to figure this out, they had to watch individual families of cells over time using a highly sophisticated genetic tracing technique. It's called MBM.
Right, I saw that acronym all over the paper. Mosaic analysis with double markers.
That's the one.
How does a double marker let you watch a cell family?
So MADM is essentially a really clever genetic editing trick. It allows scientists to permanently tag a single dividing stem cell with a specific fluorescent protein, say glowing green. Crucially, because that tag is integrated into the cell's DNA, every single time that cell divides, its daughters, granddaughters, and great-granddaughters, they all inherit that green glow.
Oh, that is so cool. It literally paints a family tree.
It does, a permanent visible family tree inside the living tissue. So researchers can see exactly how many descendants a single ancestor produces and, what kinds of jobs those descendants eventually take on.
And the specific ancestors they were tagging were called radioglial progenitors, or RGPs.
Yes, RGPs are basically the ultimate workhorses of the developing cortex. Now, in a real living embryo in vivo, RGPs follow an incredibly strict, orderly, linear sequence.
Like a schedule.
A very strict schedule. We call it temporally stereotyped. So phase 1.
Yeah.
They just multiply. They divide symmetrically to expand the pool of stem cells.
Okay, because you need a massive foundational pool before you start building specialized structures.
Right. Makes sense, right? Multiply the workers before you build the building.
Yeah.
Then, phase 2. At a very defined, coordinated moment, that whole pool switches into an asymmetric neurogenic phase.
Meaning what?
Meaning every time an RGP divides, it makes one copy of itself to maintain the pool and one brand new neuron. And they churn out these neurons in a very specific order, from deep layer neurons to upper layer neurons.
Okay.
finally phase 3. Once the neuronal architecture is complete, they switch one last time and produce glia. Those are the support cells of the brain. So expand the pool, make neurons, make a glia. 1, 2, 3.
1, 2, 3, got it. But when Stuffer's team watched those glowing green families inside the laboratory organoid, that beautiful 1, 2, 3 sequence completely fell apart.
It dissolved into absolute chaos. I mean, the families completely lost their predictable sequence. About 1/3 of these RGPs just jumped the gun.
Jumped the gun how?
Well, instead of spending the necessary time expanding the stem cell pool, they immediately started producing neurons.
Skipping phase 1 entirely.
Skipping it entirely. And because they started too early, they became restricted. They produced a very limited range of cell types instead of the broad diversity the brain actually requires.
Well, what happens to the structure of a mini brain when 1/3 of the cells just skip the multiplication phase? That sounds disastrous.
It is. You end up with overlapping, uncoordinated behaviors. You have some RGPs still trying to multiply while their immediate neighbors are already churning out late-stage neurons.
Oh, wow.
There are no clear temporal boundaries. The cellular architecture just becomes stunted and disorganized.
This feels like a massive contradiction, though.
Right?
Like we have Faravelli proving that the cells remember their age down to the chemical padlock,
Yes.
and then Stauffer showing that the cells are jumping the gun and completely ignoring the developmental schedule. How do those two biological realities exist in the same dish?
What's fascinating here is that they seem contradictory, but they actually highlight a fundamental rule of biology. Internal programming is only half the equation. The Faravelli study shows us that organoid has the sheet music. The epigenetic clock ticks away autonomously. But the Stauffer study reveals that a complex symphony cannot play itself.
Right.
Without the environment of a real body, the organoid lacks the coordination to tell every single cell what to do at that exact moment. It has the sheet music, but it is completely missing the conductor.
Okay, so if the cells are out of sync because they are missing the conductor of a living body, what happens if we actually drop them into a living symphony?
Well, that is exactly what our third source does.
Right. The Riva study from 2022, this one blew my mind. They took human cortical organoids and transplanted them directly into the somatosensory cortex of newborn rats.
Yeah, this represents a monumental leap in neurobiology. They're attempting to bridge the gap between in vitro in the dish and in vivo in the living body.
And they chose newborn rats specifically, right?
Yes, specifically between postnatal days 3 and 7, because the rat's own brain circuits are still forming at that point.
Ah, so it's more welcoming to new tissue.
Exactly. The environment is highly plastic, which theoretically gives the foreign human tissue the best possible chance to graft and integrate, rather than just being walled off by the immune system as debris.
And the level of biological merging they achieved is staggering. I mean, the human organoids didn't just survive the transplant. Over a few months, they grew 9-fold in volume inside the rat brain.
It's incredible, and the mechanism driving that massive growth, it actually solves the exact problem we discussed earlier regarding the necrotic core.
Right, the starving center.
Yeah. When the organoid is floating in a dish, it has no blood. But once implanted, the human tissue released chemical signals that triggered angiogenesis in the rat.
Which means blood vessel growth.
Right. Rat blood vessels physically grew into the human organoid. They wove a vascular network that supplied oxygen and nutrients to every single cell.
Completely preventing that core starvation.
Entirely.
And it wasn't just blood vessels either. The host's immune cells migrated into the human tissue as well.
Yes. Specifically, the rat's microglia. Now, microglia are often thought of purely as immune defenders, fighting off infections.
Sure.
But in the developing brain, they act a lot more like gardeners.
Gardeners.
Yeah, they physically crawl through the neural network and prune away weak or unnecessary synapses, which actually forces the remaining connections to become stronger and much more efficient.
Oh, wow. Which explains the dramatic difference in how the human cells matured.
Exactly.
Because compared to the organoids left floating in the lab dish, the transplanted human neurons physically transformed. The paper mentions their dendrites became highly arborized. Arborized meaning branching out like a tree canopy.
That's a great visual, yeah. A dendrite is the part of the neuron that receives chemical messages from other cells. In a dish, they often look like bare twigs.
Just sad little twigs.
Right. But with the blood supply and the microglial pruning provided by the rat, those human dendrites grew longer, thicker, and sprouted a massive bushy canopy of branches.
That's wild.
They developed a much higher density of dendritic spines, too, which are the physical docking ports where synapses form. So electrophysiologically, they fired faster and processed complex signals in a way that, well, it closely matched the profile of native postnatal human neurons.
The physical integration is incredible.
Mm.
But honestly, the detail that stopped me in my tracks was the sensory connection.
Oh, yes.
Because the human tissue was implanted in the rat's somatosensory cortex, the area that processes physical touch. So the scientists, they actually puffed air onto the rat's whiskers.
They did. They wanted to see if a sensory signal could travel from a rat's biological periphery, navigate the rat's neural pathways, and successfully cross the boundary into the transplanted human tissue.
And it did. The human cells lit up. They actively processed the sensation of the rat's whiskers being touched.
Genuinely astonishing.
But then they went even further. They reversed the flow of information using optogenetics. Now, they had genetically engineered the human cells to be light-sensitive beforehand.
Exactly.
And when they shone a blue laser into the rat's brain, it forced the human cells to fire. And by pulsing that light, they could actually drive learned, reward-seeking behavior. They taught the rat to go lick a water spout, purely by stimulating the human mini-brain.
Yeah, to clarify the mechanism there, optogenetics introduces a light-sensitive ion channel into the human cell membrane. So when the blue light hits it, the channel just snaps open. Sodium rushes into the cell, and the neuron fires an action potential.
Boo.
Boom. And that electrical signal traveled out of the human graft, down into the rat's motor circuits, and initiated the physical act of licking.
Okay, I have to pause here. Because here's where it gets really interesting. But wait. I am looking at a scenario where a human mini-brain is growing inside an animal, it is physically feeling the animal's whiskers, and it is driving the animal to seek water.
Right.
I have to ask the question that is inevitably on the mind of anyone listening to this. Does this mean the organoid is experiencing this? Like, is there a tiny human consciousness trapped in there, driving the rat like a pilot in a cockpit?
It is the most vital, ethical, and philosophical question we can ask when discussing this frontier of science, truly. But based on the biological evidence, the answer is a firm, unequivocal no.
Walk me through the why on that. Because from the outside looking in, a piece of human brain controlling behavior sounds dangerously close to consciousness.
I understand why it feels that way, but it requires looking closely at the mechanical reality of the circuit. The organoids did not feel, they did not think, they did not learn independently, and they did not experience qualia.
Qualia being like the subjective sensation of being alive.
Yes. They acted entirely as integrated biological hardware.
Okay, hardware.
Imagine swapping a processing chip in a complex machine. If you take a highly advanced sensor chip and wire it into the circuitry of, say, a simple robotic arm, the arm might react to stimuli more efficiently.
Sure.
The chip is certainly processing the signals it receives from the camera, but the chip itself isn't aware it's attached to an arm. It isn't independently deciding to move.
So the human cells are essentially just a really fancy piece of copper wire, bridging gap A to gap B in the rat's brain.
That's one way to look at it. They are a highly sophisticated structural component. When the researchers stimulated the pathway with light, the signal traveled down the line and triggered a physical response the rat had already been trained to execute.
Ah, the rat already knew how to do it.
Right. It is a stunning demonstration of biological compatibility and synaptic integration, absolutely. But it is not a demonstration of emerging consciousness or independent agency.
Okay, that distinction is incredibly helpful. Thank you.
Yeah.
So bring this back to our central mystery. We have this human organoid that tracks its molecular age perfectly, but suffers from chaotic, disorganized cell behavior when it's left alone in a dish.
Yes.
If we take it out of the dish and plug it into a living rat providing the blood, the microglia, the sensory input, does this transplant completely fix the cellular timing defect we saw in the stuffer study? Like, do the cells stop jumping the gun?
This raises an important question, and this is where we have to be very careful to distinguish between an observed fact and a biological hypothesis.
Okay.
Let's look at the conductor metaphor again. The living brain provides an immense symphony of cues, right? True metabolism, neighboring diverse cell types, physical mechanical pressure from the skull, a complex extracellular matrix.
The works.
The works. The Riva study definitively proved that providing those missing signals via transplantation caused massive advanced maturation.
In fact, it matured the cells so well that they were able to reveal structural defects for diseases that usually hide in early development, right?
Exactly. They modeled Timothy syndrome, which is a severe neurodevelopmental disorder caused by a defect in a calcium channel.
Okay.
Now, when you grow Timothy syndrome organoids in a dish, they often look relatively normal because the cells just never mature enough to rely heavily on that specific calcium channel.
So you can't even see the disease.
Yeah. Right. But once transplanted into the rat, the human neurons matured to the point where the genetic defect finally manifested in abnormal dendrite growth. The living environment forced the cells to grow up, which finally revealed the disease phenotype.
Which is a revolutionary tool for figuring out how to treat those diseases.
It really is.
But back to the timing chaos. Did the transplant fix the 1, 2, 3 sequence of the stem cells?
The honest answer is, we do not know for sure.
Really?
Yeah. The RIVA study observed advanced maturation and synaptic integration, but they did not track the minute-by-minute family tree of the dividing stem cells. We hypothesize that being in a living system might restore that strict temporal lineage sequence. It makes logical sense that the environmental cues would force the radial glial progenitors back into line. But biology is incredibly complex.
Because Stouffer looked at mouse cells in a dish, and Riva looked at human cells in a rat. So we're kind of comparing different experimental setups.
Precisely. To definitively prove that the living environment acts as the conductor that cures the cellular chaos, a lab would need to run that exact same MADM fluorescent lineage tracing on a transplanted organoid.
Oh, I see.
They would have to watch the glowing family trees inside the rat to see if the overlapping premature neuron production is actually cured by the in vivo environment. That remains the frontier of this research.
It is the ultimate biological puzzle. You have this molecular metronome relentlessly ticking away inside the DNA, locking down genes with chemical padlocks, counting the days and the years regardless of where the cell is.
Yes.
But the players in the cellular orchestra still need the environment of the concert hall. The blood, the pressure, the immune cells, to know exactly when to play their specific notes.
Untangling those two realities, the unyielding internal clock and the absolute necessity of external environmental cues, is the great challenge of modern developmental neuroscience.
Which brings us full circle. I want to take you back to the image we started with. Picture that 5-year-old human organoid sitting in a lab dish. Its epigenetic software is silently updating.
Ticking away.
The cells remember their age, they remember the stages they've completed, and they fiercely protect that memory, even while they struggle to organize themselves into a coherent structure without the physical context of a body.
It is a profound testament to the resilience of our biological programming. The drive to develop, to mature, it just persists even in a void.
And understanding this timing is the absolute key to understanding how our own brains built themselves from a single cell into the most complex structure in the known universe. It's the key to understanding what happens when that biological assembly line gets out of sync in neurodevelopmental diseases, and ultimately how we might one day design targeted therapies to fix the clockwork.
And as we inch closer to that reality, I'll leave you with a final thought to mull over. We've talked extensively about the missing conductor, right? The exact environmental signals, the blood vessels, the sensory inputs that keep the developing brain's cellular orchestra in perfect time.
Right.
If scientists eventually identify every single one of those signals, and they learn to perfectly recreate them in a laboratory incubator, can they responsibly do so? Where exactly is the ethical line when a lab-grown biological model stops being a chaotic clump of cells and starts ticking in perfect time?
That is a question that is only going to get louder in the years to come as this technology bridges the gap between the dish and the living system. If you enjoyed today's deep dive into the secret clock of the brain, please like the video and subscribe to the channel. It really helps us bring you more of these deep dives into the frontiers of science. Thanks for exploring with us, and we'll see you next time.